In this video, we're going to discuss the evidence for evolution that Darwin proposed. So Darwin proposed a theory of evolution by natural selection, and we're going to see what evidence exists to show that the theory is actually the best explanation for the diversity of organisms that exist today on Earth. So the main pieces of evidence that we're going to see are evidence that can be found in the fossil records, geographic distribution of species, comparative anatomy and comparative development, as well as molecular biology, and then also evidence for natural selection, which would fall under antibiotic resistance and artificial selection. So we'll start off with looking at the evidence that we can find in the fossil records. A fossil is just a remains of organisms that are buried in sediments. And so people who study fossils are called paleontologists. And what we have is a fossil record. So a fossil record is essentially a chronological order of organisms that existed, and we can see that chronological order by looking at where the fossils are found. So deeper in the sediments, deeper below ground here, underground, would be the older fossils, and as you go up, you'd have the more recent fossils. And so we can figure out the age or the relative age of fossils, how old a fossil is compared to another fossil, based on where it's found in the sediments. If you find something deeper, it's older than if you find something that's higher towards the top. And so what fossils have allowed us to see is, first of all, that there's some organisms that existed in the past that look very similar to organisms that exist today, which is a great support for descent with modification. But it also allowed us to see that organisms could actually go extinct, so they're not static like the church thought at one point in time. Another interesting thing that fossils allowed us to see is a transition between different groups of organisms. And let's go take a look at that in some more detail. These are called transitional fossils. So here we have the Archaeopteryx fossil. It looks like an intermediate between reptiles and birds, which goes to show that birds and dinosaurs shared a common ancestor with each other. You can see that it has feathers like birds do, but it has teeth and claws and a bony tail like reptiles do, which shows a link, an intermediary link between the birds and the reptile groups. And again, that shows that birds and dinosaurs have this common ancestor with each other. So you can see that over here we have a bird, we have the Archaeopteryx, and this is that transitional fossil, the intermediate link between reptiles and birds, and that just goes to show that we share a common ancestor with, that birds share a common ancestor with reptiles, with dinosaurs, back in the day. So you can see again the feathers, you can see the bony tail from a reptile, which is an excellent source of evidence to show that there was some common descent and some relationship between them. Here we have another example of a transitional fossil. You can see the amphibian and the fish. They share a common ancestor, and the reason we can see that is because in this transitional fossil, the Acanthostegas, we have gills like fish, and then we have lungs, legs, and limbs like amphibians, showing that amphibians and fish shared a common ancestor with each other. So it's another great intermediary link between between two different groups of organisms to show that they do share that common ancestor, and that they did experience changes in different environments, and as a result have become different from each other. Another interesting piece that transitional fossils provide us is the slow transformations of populations from land dwelling to living in the water. So here we can see the common ancestor, an organism that has a common ancestor with the whales. At one point in time, it lived on land and had, well, legs to walk around on land. But over time, as we take a look at these different transitional fossils, we can see that the legs have slowly become smaller and smaller and smaller to the point where they're no longer existent. And so now we have our whale that survives in the water but no longer on land. But the weird thing is that if you actually examine the whale structures very closely, it still has a pelvic bone where you can actually insert legs. So it still has leg sockets, basically. And why would they have that if they don't use it? Well, chances are it came from the common ancestor that used to live on land. And so if you take a look at this cladogram here, you can see that the whale and hippopotamus they're actually very closely related to each other by a common ancestor. It just so happens that some of the organisms from that common ancestor decide to live on land, and the selective pressure made it so that they would keep their legs, whereas others maybe went to live more in the water, and so if they had legs, there may not have been a selection for legs, and so they would have died off, and instead it would have been a selection for no legs and other features that allow them to swim and survive in those watery environments. Darwin studied fossils as well, and he found that some of the fossils he discovered were giant versions of present-day organisms, so they look very similar to present-day organisms, and that just again goes to show that they may have been related by a common ancestor. Another piece of evidence that Darwin used to support his theory of evolution by natural selection is biogeography. Biogeography just basically means where species are distributed on Earth. Now, before we go into more detail on that, I need to discuss a few terms, the terms continental drift and Pangea. So at one point in time, all the continents were connected together in a supercontinent called a Pangea. And over time, there was a slow movement of the continents where they moved away from each other, and that's called continental drift. And that continental drift is still occurring today. So here's what Darwin was able to explain with the idea that continents move away from each other. So here we have an island, island A, and it has one species on it, species A. And over time, continental drift occurred, and island A became two islands. Species A stayed on one portion of the island and was also on the other, but because of different mutations and different selective pressures over time, species A became a little different from the original species A. It now became species B, and they were their own species due to having a different island with different selective pressures and different mutations that occurred in them. And then more continental drift happened, and more islands were created, and so some of the original species B were preserved, but some of them became species C due to different mutations and selective pressures. And that happened over and over again with mountain ranges and more continental drift. So the main idea is that all these organisms are related to each other. They all came from an ancestral species called species A. And so A and B, or some common ancestor before that too. So A didn't come out of nowhere, it had a common ancestor somewhere. So the main idea is that A is related to species B. And then B gave rise to C somewhere along the line, and so they're closely related to each other. But they're still related to species A deep down. And then C gave rise to D, so they're closely related to each other. And I'm not saying C transformed into D, I'm saying that there were mutations, some changes, and over time, slow changes led to species D. But the main point is that C and D are related to each other, but they also initially came from B, and they all initially came from some common ancestor in A. So all these organisms are related to each other, but the reason that they became so different is because those continents, those regions of the land separated from each other, and they had different mutations, different selective pressures. They were no longer mating with each other since they were found in different regions, and as a result they could not stay similar and became so different that they became new species altogether, and they could no longer mate with each other, and they were their own different group of organisms. So one thing that scientists found puzzling is that in different areas of the world, there are organisms that look similar to each other, even though they don't really share a common ancestor or a recent common ancestor. So all organisms share a common ancestor, but in terms of relationships, how closely related you are, you have to share a recent common ancestor in time. So these organisms here, they all look very similar to each other, but if you study their DNA, they're distantly related. They're not super closely related to each other. But the reason they look so similar to each other is because they have similar environmental pressures in the areas that they live, and that's why they end up looking similar. It's kind of like saying if you see two people in Canada during the winter wearing winter coats, you can't really assume that they're closely related to each other. They might just be wearing winter coats because it's cold outside, and so they have similar environmental pressures, and that's why they both look similar on the outside, because it's cold outside. So let's go over that concept with a little bit of an animation here. Here we have a mainland. The mainland is the original land. Here we have island A. It's not an island yet. It's still part of the mainland. And these little rectangles represent, let's say, green species. And so 2 of those decide to go just hang out on island A, which is again not an island yet, but eventually it will become one through continental drift. And so that island's separated from the mainland, and I want you to pay attention to the color coding. The green represents environmental condition green, the red represents environmental condition red. So there's different environmental conditions on the mainland versus the island A. If you're green on the mainland, that's good because you can survive and hide from your predator. If you're green on island A, you probably won't survive. You can't hide from your predator very well. So over time, there may have been a mutation that led to some red organisms being produced, and they were able to survive, and the green ones died off. Now, if you were to ask somebody which organisms are more closely related, island A to mainland or island A to island B, many would say, well, island A and island B look very similar to each other. They're all red. But if you were to actually study the DNA, you would see that mainland organisms have more similar DNA to the island A organisms. The only reason that island A and island B look similar to each other is because of similar selective pressures. But deep down, island A organisms and mainland organisms are more closely related to each other because they were from the same species initially. It's just that when the islands separated, mutations occurred, different selective pressures occurred, and red was just a better color for those, and they were the ones that survived overall. So sometimes organisms can look similar to each other, even though they're not closely related to each other. You always have to pay attention to that. You have to look at the environments as well before you make a judgment or a conclusion about which organisms are more closely related to each other. So Darwin was able to actually come up with, using his theory of evolution through common descent and natural selection, he was able to propose many hypotheses, many guesses. And those guesses or hypotheses, they were actually supported by the observations he made when he went to study the chain of islands on the Galapagos Islands. And so what I want you to do is read up on his hypotheses and see how they were supported by the observations. I'll show you a few of those on the next slide. So in this table, you're going to see the hypotheses that Darwin came up with, and then you're going to see how his observations actually supported those hypotheses. So one of the hypotheses he made was that unique species are descendants of ancestral species from the nearest continental and land masses and will exhibit some similarities. So when he went to observe the Galapagos Islands, he found that there were unique species on each of the islands, but they looked very similar to species from the nearest mainland, kind of like the example I was talking about with the mainland island A and island B. And so his observations supported the guess that he made there. Another interesting hypothesis Darwin had was over time ancestral species have evolved into new geographically isolated species. When he went to take a look at the Galapagos Islands, he noticed many unique species were found nowhere else on Earth. So there were unique species on the islands that were found nowhere else on Earth. So they had evolved into unique species that you couldn't find anywhere, which essentially supported the idea that the hypothesis that Darwin had come up with. So these are a few other hypotheses over here. You should take a look at those and see how his observations supported those hypotheses. And this just goes to show why Darwin's theory of evolution by natural selection is so powerful. He's able to make these guesses, these hypotheses, using his theory of evolution by natural selection and all the rules he kind of came up with. And when he goes out into the environment and makes observations, his guesses have been supported time and time and time again. So you can't say that evolution is just a theory, because a theory is much more than just a guess. It's an explanation of the world that's supported by observations over and over and over and over again. So another piece of evidence, so we saw biogeography, we saw transitional fossils, or the fossil record, is comparative anatomy, more specifically a term called homology. Homology is a term that's used to describe similarities that organisms might have, and that similarity has to be due to common ancestry. So organisms are similar because they're closely related to each other. They have a recent common ancestor. And so if you were to study the human hand, the flippers of the harbor seal, and the gray whale, On the outside, they look very different from each other, and that's because they have different environmental pressures. Human hands are used for grasping, and these flippers are used for swimming in the water, and so water would select for flippers, not for hands. But on the inside, if you were to cut open the human hand and cut open the flipper of the whales or the flipper of the whales or the seal, you would see that the bone structure is very similar to each other. Very similar bone structures, very similar layouts, similar design, and that similar design is due to common ancestry. So the human, the seal, and the whale, they all have a recent common ancestor. They look different because of different selective pressures on the outside, but on the inside, skin deep, they're very similar to each other, and this is an example of homology being similar due to common ancestry. The function can be different on the outside because of different selective pressures, but on the inside, you are related to each other. And so comparing the anatomy on the inside, the underlying structure is something that we call homology. So here we can see the forelimbs of different organisms, human, cat, whale, and bat, and we can see the color coding illustrates a similar bone structure, and that's due to common ancestry, despite having differences on the outside, which is due to, again, different selective pressures. So why do these organisms end up looking so different from each other? Well, we call this divergent evolution. So here we have, let's say, an ancestral species. This is a species that may have arisen from a common ancestor. This might have been on the mainland. And over time, some of the mainland species flew to an island with buds and fruits, some flew to an island with grubs, some to an island where they had to use tools, some to an island with insects, and some to an island with leaves. And on those different islands, there were different selective pressures, and so those that had this shape of beak were able to survive and eat the buds and the fruit. Those that had this shape of beak could survive and eat the grubs. So on the outside, they all look very different from each other because of the different external pressures that they had to face, the different environmental pressures. The differences initially arose due to random mutations, and it just so happens that on these different islands, this was a better one, this was a better beak, this was a better beak, this was a better beak, and this was a better beak. So we call that divergent evolution, where closely related organisms just start to look different from each other. They start off looking very similar, let's say at this dot here, but over time, due to different environments, they start looking very different from each other. However, if you were to analyze their bone structures and analyze their DNA, you would see they are still closely related to each other, but they just diverged from each other in terms of looks because they had different environmental pressures. And so we call that divergent evolution, divergent meaning to move away from each other in terms of looks, but they're still closely related deep down. Another interesting homologous feature that you can sometimes study, and I mentioned this in the previous slide with the transitional fossils, are structures called vestigial features. They're essentially leftovers of the common ancestor. They're useless structures that that organisms no longer need, but they're still found inside of them. So for example, a snake and a whale, they'll have sockets for legs, even though they don't have legs and they don't need legs in their environment. So we say that those were leftovers from their common ancestor that existed in the past. And so this is an example of another homologous structure, more specifically a vestigial homologous structure. So we can see the example here again with the leg sockets on whales, and whales don't have legs, but their common ancestor did. We can see over here the dog, the pig, and the horse. They have these little bony structures on their legs that they don't even use. So these little toes on the digits on the feet of the pigs are not even used by the pig. On the dog over here as well, or on the horse, are not even used by those organisms, but they existed in the common ancestor. So they're not used anymore, but they existed in the common ancestor, so they're considered vestigial. Over here we can see a fish found in a deep sea cave where there's no light, but it still has the structures for an eye. The eye actually can't see anything. It's a useless eye, but it probably exists there because a common ancestor of this fish had that eye. And it just so happens that some of the fish maybe went to live in a deep sea cave. There was a mutation that led to blindness, but it wasn't selected against because those fish didn't rely on seeing anyways since they were in a dark cave. Whereas the fish that still relied on seeing, well, if they did go blind, they would have been selected off because they need that light to see and survive. Humans also have vestigial structures. Here's a great video that illustrates some of the vestigial structures that humans have. One great example is something called the palmaris longus. If you put your thumb and your index finger together and push up against your other hand, you might see it appear on your wrist. Some people do not have palmaris longus while others do. And so this is thought to be a leftover remnant of a common ancestor when organisms, our common ancestor, lived in trees or climbed trees more frequently. Today it doesn't really provide any use for anything. It doesn't give you any more grip strength. In fact, a lot of doctors will actually remove this palmaris longus tendon if you have carpal tunnel syndrome, to make more room for your for the tendons in your wrist. But again, it's a neat fact, or a neat piece of evidence that shows that we come from a common ancestor that might have used that in the past. Another great homologous structure you can study is something called embryological development. So this is called comparative development instead of comparative anatomy. You can compare the embryos, the developing fetus of different organisms, and what you'll see is striking similarities between the embryos of, for example, humans, pigs, reptiles, and birds, as they're all closely related to each other. We're studying vertebrate embryos here, so organisms with a spinal cord. And if you study especially the early embryos, you'll see they all have very similar structures. So they'll all start off with a tail. They'll also all have essentially gills or pharyngeal pouches. Now, as we develop, those structures either disappear or become different things. So the pharyngeal pouches would become gills in fishes, but then in other mammals it might become ears and throat in humans. But the reason that humans and all these organisms share similarities in the embryo is because, well, we all have a common ancestor, and we all started developing in much a similar way, but then as we develop and become and get older, those structures become different, and they're more easily differentiated. So we can see here in this picture the human, chicken, salamander fish, so on and so forth. Earlier on, especially early in development, they look extremely similar to each other. It's very difficult to tell these organisms apart, but then as we develop and become older, we can tell them apart. But there's still some striking similarities between all these organisms as we develop in the embryo. And that's another example of a homologous feature that we can study to show that close relationship between the organisms. So one pitfall that evolutionary biologists often face is making a mistake in terms of relationship just because two things look similar to each other. So if we look at the shark and the dolphin, they look very similar to each other. And a lot of people might say, well, because the shark and the dolphin look similar to each other, they're more closely related to each other than, let's say, a dolphin is to a cat. In reality, it's the other way around. In reality, a dolphin is more closely related to a cat than it is to a shark. But the reason that the shark and the dolphin look so similar to each other is because they have similar environmental pressures. They both live in water. However, if you were to cut open a dolphin and a cat and look at their underlying bone structure, you would see, based on homology, that the dolphin and the cat are more similar to each other. They just look different on the outside because they have different selective pressures. One is on land, one is in water. If you were to study the DNA of a dolphin and a cat would be more closely related to each other than a shark and a dolphin. Again, the only reason that the shark and dolphin look similar is because of the similar environmental pressures. That's like the example I gave earlier. Two people might be wearing a winter jacket, not because they're closely related, just because they both live in an area where it's cold. So when two organisms that are distantly related, so not closely related to each other, start to look more like each other because of similar environments, we call that convergent evolution. The similar structures that they have due to the similar environment, those are called analogous structures. So they're similar not because they're closely related, but because of similar environments. Homology is when organisms are similar not because of similar environment, but because of close relationships, common descent. So analogous structures, you have to be careful with that, because if you don't study it carefully, you might mistakenly assume that two things are super closely related to each other, even though they're not. They only look similar because of similar environments. So convergent evolution is the opposite of that divergent evolution we discussed earlier. Here we can see an example of the bat and the dolphin. They both use echolocation to communicate and find prey, for example. But here we have a submarine, and it also uses echolocation, uses sonar to locate things in the environment. But just because this submarine here uses echolocation, it doesn't mean it's closely related to a dolphin or a bat. It was just designed that way for the function, for the environment that it's in. So that's when you have to start using your knowledge to decipher relationships. So if we were to look at a bat and a bird, they both have wings. And so you might think they're very closely related to each other. But in reality, they're not. The reason they both have wings is because of similar environmental pressures. If you were to study the DNA and the underlying structures, the bone structures of bats and mice and birds and crocodiles, you'll see that bats are actually more closely related to mice than they are to birds. And birds are more closely related to crocodiles than the birds would be to the bat. So just because two things have wings, it doesn't necessarily mean they're very closely related to each other. You need to really study the homologous features and not the analogous features. Analogy is due to similar environments. Homology is due to common ancestry. The more recent your common ancestor, the more closely related you two are. This here is a summary of analogous structures versus homologous structures. Analogous structures, again, they typically will have the same function because of similar environments. So all these organisms here, their fins and flippers and wings, they have similar features on the outside because they all sort of live in the water and all have similar selective pressures. However, if you were to study their underlying fundamental structure, their bone structure, you would see they're drastically different from each other, and that's because they don't share a recent common ancestor. They have a common ancestor, but it's not extremely recent. So the shark and the dolphin may not share the most recent common ancestor out there. The only reason they look similar is because of convergent evolution, essentially having similar pressures that led them to look similar in their similar environments. Homologous structures, on the other hand, they have different functions typically, but if you were to cut them open and take a look at their underlying fundamental structure, you'd see a very similar structure, very similar organization of the bones, for example. And these similar underlying structures are due to homology, they're due to divergent evolution. That's the reason they look different, because they're in different environments. But on the inside, they're similar because they have the same, they have similar common ancestors, recent common ancestors. So you should be able to answer questions like this now, so you should pause the video and try it out yourself, and then you can compare with my answer. But over here we can see the flying squirrel and the sugar glider have very similar structures superficially on the outside, since they live in similar environments. Now their underlying structures, if you were to open them up, their bones and their muscles, they're very different from each other. So why do they have these similarities? Well, you should have been able to guess that the reason they're similar is because of analogy. They have converged to look like each other because they might have similar environmental pressures. So you don't want to, again, make a mistake by looking at the external features only and the functions only. You need to look at the underlying structures of homology, the embryological development, the underlying bone structure, and in today's day and age we actually have better technology to see even more precise types of homology, and we'll see that in just a little bit. So another more precise type of homology you can use is actually using DNA homology or molecular biology homology. So you can actually compare the DNA similarities and protein similarities in organisms to see how closely related they are. And the idea is that the more DNA similarities you have, the more protein similarities you have, the more closely related two organisms are. Now, one really cool piece of evidence to show that all organisms share a common ancestor very far back is that we all have the same genetic code. We talked about the nucleotide letters. Well, that code is read the same way in every single organism. So, well, with some exceptions. But the idea is that if I take a red pigment gene and put it in bacterial cells, banana cells, clover cells, frog cells, all of our cells will read that gene the same way as a red pigment gene. So the fact that the code is universal, read the same way by every organism, is just further proof or evidence that we have a common ancestor and we just kept on descending and acquiring differences due to mutations. So even if you look at bacteria and humans, they have very similar genes, and they read the genetic code the exact or similar ways. That is evidence that we are related to each other by a common ancestor. So we'll take a look at some examples of this. So over here we can see a picture of a human, and we're comparing it to different organisms. And we're looking at specifically a protein in the body called hemoglobin, which is found on your red blood cells. And the idea is that the fewer differences that there are between the human and the organism you're studying, the more closely related it is. So you can see that between the human and the gorilla, there's only one difference in the amino acid, which is a piece that makes up the protein. And so the human and the gorilla are very closely related to each other compared to the human and the rhesus monkey. The rhesus monkey and the human have 8 differences between each other in the hemoglobin chain. Or if you look at the human and the frog has 67 differences compared to with the human. So the frog is not as closely related to the human as the gorilla would be, or as the rhesus monkey would be. So if you were to make one of those phylogenetic trees, what you would have, and I'm not going to draw all of it right now because we'll see this in more detail later on, you would have a frog being distantly related to the human, and then you would have over here the human and the gorilla being very closely related in time and in common ancestry over here. And then the other organisms would be somewhere in between here. But that's all based on the protein differences. The reason we can use protein differences to determine how closely related organisms are is because the proteins are coded for by the DNA. So the idea is the closer your proteins are in structure, the closer your DNA must have been, because that's the instruction to make the protein. So here we can see an evolutionary tree, or a phylogenetic tree, or also called a cladogram. It is a picture that shows how closely related organisms are through common descent. So again, if organisms share a recent common ancestor, it means they're closely related to each other. So for example, the turtle and the leopard are closely related to each other. In fact, the leopard is more closely related to the turtle than it would be to the salamander, because yes, it shares a common ancestor with it over here, but with the turtle, it shares a more recent common ancestor. All these organisms are related to each other, all the way back here. For example, you can see the common ancestor here, but the more recent the common ancestor is, the more closely related they are to each other. And the way we built this tree is by using, well, homology evidence. We can't use analogy evidence, we have to use homology evidence. And so that homology evidence can come from DNA, it can come from proteins, it can come from other structures we discussed, like bone structure. So over here we decide to use these homology pieces here. These are called characters. We used hair, amniotic egg, 4 walking legs, jaws, and vertebral column. And on the top over here you can see the different organisms. This is called a character table. If the organism has that character, there's a 1 written in a box. If it doesn't have that character, there's a 0 written in a box. So for example, the leopard has hair, amniotic egg, 4 walking legs, jaws, and vertebral column, whereas the turtle doesn't have the hair, but it does have the amniotic egg, legs, jaws, and vertebral column. And so the idea is that the more characteristics 2 organisms share in common, the more closely related they are to each other. So the leopard and the turtle share 4 things in common with each other. The leopard and the salamander share 3 things in common with each other. The leopard and the tuna share 2 things. The leopard and the lamprey share 1 thing. The leopard and the lancelet share 0 things with each other, at least of the things that we're studying. But deep down they might share other things. So what we would do is we would connect them in that relationship. Leopard and turtle share the most things, so they're more closely related to each other. Following that would be our salamander, because it shares 3 things with the leopard. Following that would be the tuna, because it shares 2 things with the leopard. Following that would be the lamprey, because it shares 1 thing with the leopard. And following that would be the lancelet, because it shares, well, 0 things with the leopard that we're studying in this table. And then you can place where the different characteristics arise. So for example, the lancelet, that's called our outgroup. It does not have anything in common with any of the other organisms. It's still related, but just very distantly related. We use it as a group of comparison. So the lancelet doesn't have anything, but the first thing to sort of develop is the vertebral column, and so we could put that over here. And that means any organism after the vertebral column has it. So lamprey have it, tuna have it, salamander, turtle, and leopard. Jaws, well, the lamprey doesn't have it, so we have to put it after the lamprey's common ancestor. But everything after the lamprey has it. So the tuna, salamander, turtle, and leopard have jaws. And so we can keep placing the traits as they developed. For example, you can see the leopard is the only one that has a hair, so we can put the hair after its common ancestor, but we wouldn't put it after the turtle because the turtle doesn't have the hair. And so we can use these homologous features. DNA is not on here, but that could be something else we use too, in order to see how closely related organisms are and help us to build these trees to show common descent. Here we can see a tree that was built using differences in protein structure. So again, the human versus a rhesus monkey over here, very few differences, so they're very closely related to each other. A few more differences between the human and the mouse, so still related but a bit farther back. More differences between the human and the chicken, so still related but even farther back. And then even more differences between the other organisms. So they're all related to each other, but just farther back in time, the more differences there are between the organisms that you're studying. And again, we can study protein differences because protein are coded for by a DNA. Closer your protein are, therefore the closer your DNA would have to be to get you the similar protein structure. And so the benefit of using the protein and DNA similarities is that, well, we can avoid the confusion between homologous and analogous structures in our studies. So another piece of evidence that we use to support Darwin's theory of evolution by natural selection is artificial selection. So Darwin was saying that nature selects for certain characteristics and against characteristics, and people had a hard time believing that. But then Darwin said, this is not really weird when you think about it. Farmers do this all the time. So for example, here you can see the wild mustard plant, and farmers, for example, may have selected for a larger lateral bud. And when they did this over many generations, just kept breeding the plants with the larger lateral buds, they ended up becoming Brussels sprouts. Or if farmers selected for a larger terminal bud and they did this over many generations, they would get a cabbage. Or if they did larger flower clusters, they would get the cauliflower. So depending on what farmers selected for, they would get different looking organisms over time in those generations. So if humans can select for characteristics, why can't nature do it as well? That's what Darwin was trying to get at with the idea that artificial selection can be used as evidence that natural selection can also happen. So here we can see another example of artificial selection. Farmers selected cows with the highest muscle mass to breed over generations, and eventually we got cows with really, really high muscle mass. Over here, different breeds of cats and dogs with different sized nose and ears and tails. Those are the result of artificial selection. Humans are breeding animals with certain characteristics that they want to see more of in the next generation, and do that over and over and over again until they become accentuated or exaggerated in future generations. So artificial selection is very similar to natural selection. The main difference is that natural selection is the environment choosing which traits are passed on, which traits will become adaptations, whereas in artificial selection, it's the humans that are selecting for it. But again, Darwin's logic was that if humans can do artificial selection, well then nature can technically do natural selection. The environment can select for things too, just in a different way. Another great example of natural selection is antibiotic resistance. This is something that we observe quite a bit. So when you get sick, if you're infected by a bacterial cell, such as another type of cell, some are good, some are bad, but if you're infected by a bad one, you'll get medication called antibiotics. And those antibiotics will help to kill the bad bacteria. Now, if you don't take antibiotics properly, what you do is you lead to a population of very resistant bacterial cells. Antibiotic resistance is when bacterial cells won't get killed by the antibiotic that you're using. So let's take a look at our initial population. Here we have a population with some variation that was caused by mutation. Some bacterial cells have low resistance, so they'll probably be killed easily by your antibiotics. And others have high resistance, so they will not be killed as easily, and they might need more of a dosage. So your doctor gives you antibiotics, you start taking them, and you kill off the weak ones, and you start to feel a bit better because there's fewer bacteria in you, so you decide to stop. What you don't know is that you left some of these highly resistant bacteria alive. You did not complete your dosage of antibiotics. And so now what happens is that these guys reproduce, and they pass on their antibiotic resistance gene, and now we have populations of resistant bacteria instead of many weak ones and a few strong ones. And so now these are harder to kill, and we often have to develop new antibiotics in order to do that, and it makes our current antibiotics less effective. So that's why it's always important to take the antibiotics completely, or to not take them at all if you don't have a bacterial infection. Some people, when they get sick, let's say have a cold, they start to take antibiotics, but that won't do anything to a virus. Antibiotics are specifically designed to kill bacteria. So now what you're doing is you're not making yourself feel better, and you're also providing a selective pressure that actually selects for the strong bacteria and not the weak bacteria, which can lead to more problems down the road with the medication that we have, making them useless, and then maybe making some kind of population of bacteria that could really kill a large proportion of our population. So again, this is an example of natural selection happening in the real world. People needed to see evidence of it, and we can see evidence of it right here when we select for antibiotic resistance by not taking our antibiotics properly. Again, antibiotics did not create this resistance. It was already in the population. It just accentuated it. It just killed off the weak ones, and the strong ones were able to survive, and therefore now we have populations that are made up of resistant bacterial cells only, or primarily. So this is evolution, a change from a mix of resistant and non-resistant to mostly resistant. And it was caused by the process of natural selection. The antibiotic was providing that selective pressure, selecting for resistant bacteria. In this picture, you can see how antibiotic resistance works. It's just a simplified example, but bacterial cells have genes on plasmids, and there could be a mutation that creates a gene that allows that bacterial cell to make an enzyme which can alter the antibiotic that comes into the cell. And so now it won't kill the cell anymore. Or it can make a mutation that leads to a gene which produces an enzyme that destroys the antibiotic, and now it's harmless to the bacterial cell. Or there could be a gene that allows a pump to be embedded in the bacterial cell membrane, wherein as soon as the antibiotic comes in, the pump grabs the antibiotic and then spits it back out. And so this just goes to show that scientists constantly have to develop new antibiotics in order to combat these defense systems that bacterial cells seem to develop. Again, I want to highlight that bacterial cells don't develop this for the purpose of defending themselves. It just happens by accident, through a random mutation. But if you don't take your antibiotics properly, you're now going to get more of these superbugs swimming around in your body and in the populations, and it just makes it more difficult for us to fight these infections over time. So to finish off, I want to discuss the idea that Darwin didn't really use the word evolution very often in his book. In fact, I think he only used it once, because in his mind, evolution implied that progress was happening. You were always getting better. But the idea behind descent with modification is that we're all related by a common ancestor. And over time, there's modifications that happen through mutations, and the environment just selects for which one of those modifications will continue surviving. And it's not saying, this one is better all the time, this one is better all the time, this one is better all the time. What the environment is really doing is it's just selecting organisms that are better for that environment at that point in time. When the environment changes, the characteristic that was once an advantage could become a disadvantage. Or the characteristic that was once a disadvantage could become an advantage later on, depending on the environment. And so this is a great book. If you ever have an opportunity to read it, I highly recommend it. It's called Survival of the Sickest. It's a book that explains how many diseases today, for example, diabetes, were once survival advantages in different environments of the past. Just goes to show that, again, evolution is not about getting better. It's about being a match with your environment. And that environment changes, and so you won't always be perfect for every environment. So what we saw is that all organisms are related through ancestor, a common ancestor. And over time, the common ancestor becomes different. The common ancestors become different because of mutations in DNA. And so if those differences are separated into different environments, well, they won't be able to mate with each other, and they'll become different species altogether. Certain differences are selected for in different environments because of different selective pressures. What I need you to understand is that natural selection is the process that caused evolution. So Evolution is a change in gene frequencies over time, from maybe having a equal amount of green and brown to having more brown than green. Or if you're looking at macroevolution, it's about new groups of species being produced, and that's with many generations of microevolution happening. But the main idea is that natural selection leads to that evolution. Some organisms die off, others survive, and we have a change in our population makeup. Now, natural selection is just one mechanism. It's not the only process that can lead to evolution. In the next part of the evolution unit, we're going to learn about a few other processes that can lead to a change in gene frequencies over time. We'll see things like gene flow. We'll see things like genetic drift. And those are all different processes that could lead to a change in gene frequencies. So what we studied in the first few presentations were patterns of evolution showing a change in the makeup of the population over time. We studied the mechanism of evolution known as natural selection. We studied what Darwin proposed as his theory, which was that all organisms are related by a common ancestor, and that over time natural selection will select individuals better fit for different environments. And the differences that arose were due to mutations in DNA, even though Darwin didn't directly use that word himself. So Darwin's theory is not just a guess, it's supported by evidence, which we saw in this presentation. We saw homology with DNA and bone structure and proteins and embryological development. We saw direct observation through observing artificial selection and natural selection as it occurs in bacterial cells. We saw distribution of species known as biogeography and hypotheses supported by observations made by Darwin in nature. And we also saw evidence that came from fossils, such as vestigial features that no longer exist in organisms, and also transitional fossils. So a few important points to finish off with is that natural selection only works on variation that exists already or that came from mutations. Natural selection does not create variation, it acts on the existing variation that's there, and it has to be heritable variation. Remember that individuals do not evolve, it's populations that evolve. So natural selection acts on the individual, but overall that'll change the population. So populations evolve. And again, natural selection does not lead to perfection. It just selects against or selects for certain traits that already exist there. It doesn't make the perfect being. It doesn't shape the perfect being. It just acts on what already exists there. It only works with what it has to work with. The readings you can do are found on these pages here, and these are great questions that you can work on. There are some great videos you can watch that summarize the evidence of evolution along with evidence of evolution in the human body. You should also work on the guided reading questions that you'll find on the course website, or you might already have a hard copy of it as well.